Okay, so today we're going to be talking about topic 2.5, which is all about enzymes. So in 2.4, we figure out that amino acids are linked together through condensation reactions to form these polypeptides, which we'll eventually call proteins. And again, just a reminder that amino acids, we can identify them, that they have an amine group on one side and a carboxyl on the other with some sort of R group in between them. And when we have them stretched out like this is our primary structure, and they are linked together by those very special peptide bonds between the carboxyl of one amino acid and the amine group of a different one. And they can form together again in those condensation reactions to give us that polypeptide chain. And then a reminder that the amino acids can all have very different characteristics. They have different sizes, different hydrophobicity, different charges, which will affect the folding as soon as that protein is created. So we end up with these tertiary structures. In the globular proteins, we have the alpha helix and the beta sheet. And again, tertiary is when you have the entire structure, and secondary is just the local folding. So when you're looking at these whole proteins, they can have multiple secondary structures like these alpha helices. And then just a reminder that the protein takes its shape because of all of the characteristics of the amino acids, that as this chain is being created, it's starting to form hydrogen bonds between that oxygen on a carboxyl of one amino acid with a hydrogen on the amine group of another amino acid somewhere else. That those amino acids have characteristics. The hydrophobic residues in water are going to kind of want to run inside. And then those charged amino acids, the positive and negative, are going to affect the folding too. So as the primary structure comes off, it will start to fold in those secondary structures, the alpha helices and the beta sheets. And again, it's all being held together a lot by the hydrogen bonds and those hydrophobic interactions, which will eventually give it a shape. And we found out when we were doing our egg lab that we can interfere with that shape. We can denature the protein either by applying heat, just like when you cook your egg, or when we were changing the pH by adding acid or by adding a base. All of them are going to interfere with those hydrogen bonds. They can also interfere with the ionic bonds. So this was similar to when we could vaporize water by adding heat. You're basically breaking those hydrogen bonds, and when we do that in a protein, it denatures. It completely unfolds and is therefore not functional afterwards, right? So let's go back to, our metabolic reactions. Your body is constantly building and breaking things down, and this is basically chemistry, right? So chemical reactions are happening all the time in your cell, and most reactions are going to require some amount of energy to go. So if we start with some set of reactants, it will create some set of products, but typically there's some sort of hump that I have to get over. I usually have to put some kind of energy in the system in order to make the reaction go. So we call this difference between where we start with our reactants and the energy required for us to drive the reaction to get the products, the activation energy. And so most of the time it is a pretty steep hill. And what this is basically saying is that the reaction will not occur spontaneously without the addition of this amount of energy to drive the reaction forward. So enzymes are a special class of protein that lower this activation energy in order to make the reaction more likely to happen. And what is happening here in this midpoint is that we're in a transition state between our starting products and our reactants. And usually this transition state is very unstable. It's energetically unhappy. It doesn't want to do it. So what an enzyme will do is basically come along and grab those reactants and hold them in a way to stabilize that transition state. And that's what allows the activation energy to come down and for that reaction to proceed. So remember that proteins can have any size, any shape pockets in that active site. So it will fit any specific substrate specifically and hold it in that transition state to make it happier so that the reaction will actually occur. So this is like an energy diagram of how an action or a reaction would proceed. So we start with some sort of reactants and we have this transition state that requires a certain amount of energy to hold and stabilize, and then once the reaction completes, the energy comes down when we have our products. So if we look at like a sample reaction, right? Let's say we're looking at the degradation of hydrogen peroxide into water and oxygen products. So if you have hydrogen peroxide somewhere in your house, you can probably figure out that if you take your hydrogen peroxide out and you leave it like on your counter for some amount of time, it probably will decay into water and oxygen components if you just leave it there. But it's going to take a really long time for that to happen, because this reaction going from the products on this side of hydrogen peroxide to the, going from the reactants of hydrogen peroxide on this side to the products of water and oxygen will occur faster, but you need to have some sort of input of energy into that system to make that reaction occur. So there is an enzyme called catalase that will stabilize the hydrogen peroxide in this transition state and allow it to decay into the water and the oxygen much quicker. So enzymes are just a class of proteins that help speed up a chemical reaction by lowering that activation energy. And again, it's by stabilizing the transition shape somehow. And so enzymes are basically proteins with a job. They either help link things. So when we've been linking our monomers of saccharides to make the polysaccharides, when we've been linking our amino acids together to make the polypeptide, there's an enzyme that does that specifically to do those condensation reactions. So the enzymes will help to do those condensation reactions. For every condensation reaction we've talked about, there's an enzyme that's specific for it. When we want to break those macromolecules apart, there's a different enzyme that helps break those bonds by hydrolysis. So it's either building things, enzymes are either breaking them down, or they're altering them in some way by maybe putting a phosphate group on it. It's altering those substrates in some kind of way. And the enzymes always have an ACE name, so something ACE. And the first part of the name is the thing that it's acting on. So if you work at like a burger store, right, and you're making burgers, so you would be the enzyme that's making burgers, so you would be called a burger ACE, because the burger is what you are acting on, it's what you're building. So if somebody else was like a car builder, they're taking car-like parts and putting them together to create cars, so they would be like a car ACE. So enzymes have the same naming convention. So lipase is something that breaks down lipids. So we go back to our lipids that we haven't seen in a while. These are our carbon-heavy compounds. There are enzymes specifically that chew up only those lipases. So again, they're enzymes, so the active site is specific for a specific shape, a specific size, a specific charge. A lipase will only break down lipids. So sucrase, right? You can just guess that sucrase sounds a lot like sucrose, and sure enough, sucrase is the enzyme that breaks this bond between our glucose and our fructose to free them and create the monosaccharides. So there's basically an enzyme for pretty much everything that you have in your body. So again, with proteins, they can be any shape, any size, and each enzyme will only bind a specific substrate. They fit like lock and key. So each one of these proteins, you've seen how varied they are. They're going to have a different shape, a different size, and the enzyme that goes with it will only fit a certain substrate. So if I have my like circle enzyme with the pizza entrance, it will only accept pizza parts of those certain size. If this was a giant triangle, it wouldn't be able to get in there, so the enzyme would not act on it. And similarly, if I decided to line this pocket right here with positive charges, it would not accept even a similarly sized and shaped protein in there if it had also positive charges. So there's lots of ways that I can choose to exclude certain substrates. I can choose by size, I can choose by shape, I can choose by hydrophobicity, I can choose by charge. So these are really specific. It will only fit a certain substrate. So if I had a different enzyme and this pocket was square shaped and this specific size, the squares could try to get into that other pizza enzyme, but it won't fit. It'll come back out and it will only be able to access the pocket of the enzyme that's specific for its size and shape. So we call this pocket where the enzyme is doing its work the active site. So where the substrate tucks in to the enzyme is the active site. And the thing that it acts on is called its substrate. So this is an enzyme and this is its active site, and then this is its specific substrate. It will only accept that specific substrate. So similarly, this is a different enzyme with a different shaped active site, and it will again only accept its specific substrate because you can't stuff it in there, right? So if you tried to put your socks on your dog, the dog's feet are very differently shaped. They're definitely different sizes. It probably wouldn't work so great for you to try to put your socks on the dog. And similarly, if you got socks for your dog, good luck getting your feet on those because it's, again, very different shape, very different size. So same idea with the enzymes. They're built only for a specific substrate. They will only accept that specific substrate. So now let's talk about the enzymatic rates. So enzymes do drive reactions. They do lower that activation energy and they do make the reactions go faster, but enzymes are proteins, and they are still kind of beholden to the laws of physics and the laws of chemistry. They exist in the physical world, so the enzymes are going to respond to environmental conditions, right? And so before we get into that discussion, let's look at how can we even measure how fast a reaction is occurring. And so one way that we can do that is by looking at how fast a product forms, right? So let's go back to our catalase reaction where we're looking at the decay of hydrogen peroxide into its two product components. So if I have my catalase enzyme in here and I want to see how well is my catalase reaction occurring, all I have to do is measure the products. And I have my choice of two of them, right? I could measure the accumulation of water, or I could measure the accumulation of oxygen. Either one of them is going to tell me how fast the reaction is occurring. So you do have a choice there when there are multiple products. When we did the lactase enzyme lab, I asked you, hey, what is lactase? Like, what can you guess it to choose up? And if you were looking at the front of the name, you can guess that lactase is the enzyme that digests lactose, right? And so that would be correct, right? So because the names are similar, we know that lactose is this disaccharide, so lactase is going to come along and break that galactosidic bond. And again, this is a 1,4 galactoside bond. It's named that for the fact that this is galactose on the left side and that this is the 1, 2, 3, 4. So this is the 1 carbon, this is the 4. So that's a 1,4 galactosidic bond, just a reminder there. So this is the structure of lactose, and lactase will come along and break that bond and free you back into your monosaccharides of galactose and glucose. But lactase is just a protein, right? And so if we want to measure the activity of lactase, we have choices. We can either follow the accumulation of galactose or the accumulation of glucose. And what we were doing with the Benedicts, right? If it's a blue color, it's basically measuring the amount of glucose. So this is what Benedicts is telling you. If there's a red color, it means it's detecting more glucose. And if it's a blue color, it means it doesn't detect any glucose. So the trick about Benedicts is that it will not react with a disaccharide. So when we're in even though there's a glucose here, when it's in the disaccharide form, Benedicts cannot bind to it. So it only detects glucose after lactase has had a chance to come in and break that bond and give us that free monosaccharide. So this is how we were measuring the activity of the lactase enzyme. And so just a reminder that enzymes are just proteins, right? Lactase is just a protein, and so therefore it is going to react the same way that a protein would, right? So when we were denaturing the albumin protein in the egg whites, we denatured it by heating it, right? That was one way. And we also denatured it by changing the pH when we were adding the acid or making a really basic solution. Those were the factors that denatured an albumin protein. And so it stands to reason that these factors would also affect lactase in pretty much the same way because lactase is just a protein. So the lab that we did was looking at different substrates, right? So I'm saying lactase will only digest lactose, which is found in milk. And so the purpose of having tubes 1, 2, and 3 was, first of all, glucose, right? This is like our positive control. So when we added Benedicts to the glucose, this is the one that turned like bright red kind of immediately. And then the sucrose versus the sucrose plus lactase, that was meant to demonstrate that lactase will only break down the milk because it had the lactose sugar. So the sucrose and the sucrose plus lactase would have stayed blue because lactase can't act on sucrose. Sucrose has a very different shape. So there was no way that it was going to cleave that bond and free any glucose. So these two would have stayed blue. So similarly for number 4, the milk there, right? It has lactose in it, but without lactase, it wasn't going to break those bonds and free any glucose, so it would have also stayed blue. But tube number 5 would have turned orange because I had lactose in that milk, and then when I add lactase, it was freeing glucose by breaking those bonds and giving me that red color. So the hot milk, the temperature that we were using in class wasn't hot enough to denature it. So this one turned pretty orange for everybody, but all 7, 8, and 9 stayed blue for everyone because when we have cold temperatures, an enzyme reaction just doesn't really happen. And then of course when we unfold it by changing the pH, by adding either an acid or a base, lactase couldn't do its thing, so they stayed blue. So the factors that affect enzymatic rates, just like proteins, are going to be heat, the pH, and then also substrate concentration, right? Because if you put more substrate on there for it to act on, it's going to create more product. And what we're going to find out is that each enzyme has an optimum pH and temperature. It's specific, right? And usually that's because they're built for certain conditions. So if we look at this graph here, and you do have to be able to draw them and explain what's happening in here. And so we're looking at temperature. So this is activity of an enzyme against temperature. And so what's happening at this end between 0 and 20 degrees is that not much product is being created. And the reason why is that when we're at lower temperatures, and this is just physics, components just don't move around very fast. They're kind of sluggish. And so if the substrate isn't moving around very fast, it's less likely to encounter that enzyme. And as it's less likely to encounter the enzyme, the reaction is just going to proceed slower and you'll have lower amount of product. As we warm the temperatures, though, the substrates and the enzymes are moving around more, and that substrate will encounter that enzyme more frequently, and you're going to get this increase in product until we hit some sort of optimum. But after we hit the optimum, the activity starts to drop off really rapidly. And what's happening here is that, remember, this enzyme is just a protein, and one of the ways that I could denature a protein was by heating it past a certain point, because applying that heat is going to start to undo those hydrogen bonds, and then after a certain point the protein will denature, and because the enzyme is just a protein, as you hit higher temperatures it will just fall apart and then can no longer do its job. So there is an optimal temperature for each enzyme. So these are just different enzymes from different organisms, and they have a maximum. So the C whatever that says, has a maximum at 60 degrees, but that's different than this one here at 75. So this is true for every enzyme, including the lactase enzyme, which has a peak around 40 degrees, which is close to body temperature, so that makes sense. The other thing that can affect enzymatic rates is pH, right? So some of them have an optimal pH, a lot of them are going to have neutral, and this is like what you would look at if you put a protein or an enzyme in an acid solution. It starts to denature over here. If you put it in a basic solution, it's going to start to denature on that side, because again, we're breaking those hydrogen bonds by sticking hydrogens where they shouldn't go or taking them away, and the whole protein or enzyme will just completely denature. And so each one will have its own optimum pH depending on where it functions. So like our digestive enzymes are going to have a peak at around 1.5. That's the pH of our stomach. When you're eating potatoes, the potatoes are melting in your mouth because of a protein called amylase, because amylose is the starch that's in potatoes. So in your mouth, it's about pH 6, and that is where the amylase enzyme is peaking. So they're specific depending on where they are and what their function is. So each enzyme is going to have its own optimal pH and its own optimal temperature. The other thing that's going to affect an enzymatic reaction is the substrate concentration. So if I don't have many, it's not going to react that much. But if I dump a ton of substrate on it, it's going to increase and increase and increase until I have so much substrate that I start to run out of enzyme. So that's what's happening on this part of the curve. So if I have a lot of enzyme and not a lot of substrate for it to chew up and act on, you're not going to create much product. But as I add more and more product, it is going to increase in this sort of logarithmic fashion, and then it will hit a peak after I have saturated it. So this is what saturation looks like, that I've dumped so much substrate on here, there's nowhere else for it to go, and you kind of reach a maximum. So once the substrate is equivalent to the enzyme, you're not going to get any more product out of that because the reaction is saturated. So you do have to know how to draw these graphs and how explain what's happening in the different parts of them. So enzymes have been used for commercial purposes, right? So sometimes we need to run a chemical reaction on an industrial scale. Like, could we make bacteria that does this en masse? Yes, but it's expensive. They're kind of finicky. You have to feed them and stuff. So we have developed this technology where we use an immobilized enzyme that's attached to like a bead to create our products. So the classic example of this is actually lactose-free milk. Right, so you might go to the store and you see lactose-free milk. Well, they didn't genetically engineer lactose-free cows. What they did is they took normal cows and normal milk, and then they created all these beads with the lactase enzyme attached to the outside of them. And so what they do is they have their beads and they have lactase attached to those beads, and then all they do is pour the lactose on top of it, and go back. And then as the lactose is going down, it's being chewed up, and they collect the lactose-free milk at the bottom. And so then people get all of the goodiness of milk without all of the bad effects. So that's the last part of 2.5. Yeah, we'll see you next time for 2.6.